Hello, I'm glad to have you once again for this lecture on GIT micrographs explained. As I promised earlier on with the other video explaining micrographs on the esophagus to the stomach, I am taking on small and large intestine, and then we want to appreciate the micrographs. I'm Dr. Aidoo with the Department of Anatomy and Cell Biology, School of Medical Sciences, University of Cape Coast. Be my guest. The small intestine is the portion of the GIT which helps in most of the absorption. We know that it is connected to the stomach by the pyloric sphincter. We want to look at the architecture of this very important section of the GIT. It followed the general architectures in having a mucosa, submucosa, muscularis externa propria, and then either adventitia or serosa. So we take the mucosa first. And the mucosa form crypts, which are actually deep pits. So that is the crypt. So when the mucosa is thrown into folds, it comes down and rises up again. So it forms pits which go deeper to form glands. So those are the pits. Then under the mucosa, we take the epithelium, the lining epithelium. The epithelium has cells. Some of these cells are the enterocytes, which are the resident cells there. These enterocytes are simple columnar cells, and they have microvilli, which can also be described as the brush border or striated border. Interspersed among the enterocytes will be goblet cells, which produce mucus. So it is a mucus secreting cell. Note the spelling for the secretion, the mucus, and also note the spelling for the gland when we want to describe it. So one is with MUCUS, one is MUCOSA for mucosa. I think this is not even the best example I want to use. There's one that has like mucosa gland. Okay, when we get there, I'll show you. This is just a mucosa as in part of the layers. So when we get to a better description, I'll bring that up again. Then you have Paneth cells, which will occupy the deepest parts, the crypts. Remember the chief cells in the stomach? Okay. And this is the APUD cells. You should know the full name by now. Imine precursor uptake decarboxylation. Then you also have villi. You have villi. The villi are formed by the mucosa throwing the rest of its counterparts into folds, finger-like projections. So villi, finger-like projections of the mucosa. Please note the difference between the villi, its composition, and then the microvilli. So the microvilli will be on top of the villi. So that is one villus. That's another villus. That's another villus. Okay, with the mucosa core. On top of the villus are the enterocytes or epithelial cells, which will have extensions on them known as the microvilli. Then the crypts contain intestinal glands known as the crypts of Lebekun. So as I explained earlier on, the crypts are located down there in the deep pits. Glands can be described as simple tubular. And you may find some isolated lymphatic nodules which belong to the gut. We have explained this also. Gut associated lymphatic tissue. Then when these isolated lymphatic tissues become aggregated, then they become known as Peyer's patches. Okay, instead of being isolated and sparse, when they become very, very aggregated, which means that they are a lot, okay, and aggregated, we call that Peyer's patches. Then obviously muscularis externa will be inner circular, outer longitudinal, which will be intact. When we go along, you get to know that in some parts of the large intestine, you don't have them intact. So lamina propria, for confirmation, that is the loose connective tissue which belongs to the mucosa, okay, throwing the rest of the counterparts that form the mucosa, that is epithelium, and then also the muscularis mucosa are all going up. So you find crypts again at the bottom, that is submucosa. This is quite thin in terms of its width. The submucosa is quite small. Muscularis, you have the inner circular and then outer circular. So muscularis externa, they have adventitia. Move to the first part of the small intestine, which is the duodenum. We concentrate more on the submucosa because that is the striking difference between this particular section and the others. It contains Brunner's gland, which are branched tubulo-alveolar type of gland. They also secrete mucus. They secrete mucus. So that will be the region marked by the green dotted lines. So that is the submucosa. And you can see that the submucosa is quite wide in the duodenum. And it contains the submucosal glands known as the Brunner's gland. That will be the muscularis externa. Then obviously on top of the submucosa will be the mucosa. So the mucosa will be made up of the epithelium, the lamina propria, muscularis mucosa, okay, which has been thrown into folds. So again, have LP, lamina propria, that is a villus or villi, and then the crypt of Lieberkühn. That is why you see the basophilic stain. And that's the muscularis mucosa. It is not too clear at this point. Then you have the submucosa. Then inner circular, outer longitudinal, and serosa. So from the duodenum, we go to the jejunum. So again, in the jejunum, if you want to even concentrate on the submucosa, you can tell that there's a big difference between the submucosa of the jejunum and that of the duodenum. You can't find glands. There are no Brunner's gland. So just note that for duodenum, anytime you talk of duodenum, remember submucosal glands, Brunner's. Okay. All right. So now let's start our journey from the lumen all the way to outwards. So this is within outwards journey. Within outwards. When we say within outwards in histology, we mean that we are coming from the lumen and going out. So it is medial to lateral. So that is within outwards. And then without inwards means you are starting from outside and going inside. So sometimes you'll be asked to list some layers in a certain order, whether within outwards or without inwards. So if it's within outwards, we say mucosa, submucosa, muscularis, external propria, then serosa. If it's without inwards, that means you are going to start from here, subserosa, muscularis propria, subserosa, muscularis propria, submucosa, and then mucosa. So let's start from within outwards. So the mucosa has villi, as you can see. And then there will be crypts as well, all the way. So that will form mucosa. That is the muscularis mucosa, the smooth muscle in pink. Then this big region is a submucosa, which will contain most of the blood vessels that will feed the epithelium, because you know that the epithelium lacks vascularization. Then that is the muscularis propria, inner circular, outer longitudinal. Again, the profile is not the same. As you can see, one is going upwards, and then the other one is running parallel or longitudinal. At this point, you can see that the submucosa is now throwing the mucosa also into what? Folds. Okay, it's throwing it into folds. So again, don't confuse yourself with microvilli, villi, and then the plicae secularis. Okay, so the plicae secularis will have the submucosa core, okay, together with the mucosa. Then the villus will be just the mucosa, okay, and the microvilli will be extensions on top of the enterocytes or the simple columnar cells. So we now look at the portion of the jejunum. It has this characteristic appearance, so you won't miss it. Okay, you won't miss it. Very characteristic. The villi are very, very numerous and characteristic. The pattern is so beautiful. Now that will be the villi, and then you have a submucosa core, another submucosa core. So the P is standing for the plicae. So this whole extension will be a plica. One is a plica, and then the plura is plicae. So V is standing for villi or villus. So that's the submucosa, and then the muscularis mucosa. Muscularis externa, and then serosa. So again, you can see that very characteristic pattern of the jejunum. So again, muscularis mucosa is there, circular, and then longitudinal muscles, muscularis externa. Another type of micrograph, jejunum, again, submucosa going into the core, forming a plica. Okay, very, very beautiful outline. And you have some of the submucosa that didn't go to form the core of the plicae, so at its position. And the muscles, muscularis externa. You can see some blood vessels are also found within the submucosa. Again, even without writing the name, you can tell that this is jejunum. Okay, look at how their villi are oriented. So villus, plicae secularis, submucosa for SM, M is muscularis externa, S is serosa. So V, villus, P, plicae secularis, or plica secularis for one. This is also so beautiful. So that tells you that you will not miss any micrograph of the jejunum. So this is the ileum. Its microvilli are not as extensive as that of the jejunum. It also has the scripts of Lieberkühn with Paneth cells there. It also has plicae, also known as the valve of Kerckring. So again, submucosa going into the mucosa and throwing it into fold. This ileum. So this is another type of micrograph of the ileum. I like this better than the previous one because this is well stained, and it also shows one characteristic of ileum compared to jejunum. And these are lymphoid nodules. You have lymphoid nodules, as you can see there, occupying most of the submucosa. So the crypts will be here in the mucosa, and then the muscularis mucosa will outline the limits of the mucosa. So that will be also villi there. And where you have the submucosa going together with the villi, you obviously have the plicae. So between the ileum and then the cecum, there's a junction. So it means that we are now moving from the small intestine to the large intestine. So you have the ileum portion. Mucosa has villi and crypts. And then there's an extension of the muscularis externa. Usually the inner circular muscle participates prominently in the formation of the junction. Okay. So this forms something like a valve. Okay. So there's the portion here for ileum and then the portion there for the cecum. It's just like a barrier, like I used the Burkina Faso and Ghana border as an example, that you can put 1 foot here and 1 foot there. So that is it. There's a barrier there. So there's a transition from the villi and crypt-like, lining to a mucosa which has crypts only and no villi. That is the ileocecal valve, extension of the muscularis externa. So for large intestine, we are looking at cecum, ascending colon, transverse colon, descending colon, sigmoid colon, and then the rectum and anal canal. Neither villi nor plicae secularis are present in the large intestine. So that is one very, very prominent characteristic you need to take home. So that is it. You can see that there are no villi because the surface is continuous. Okay, that is the epithelial surface. Only crypts because you have the invagination, the pits. Okay, so the epithelium invaginates, goes down and comes up. It does not go up and then come down on top of its line. Okay, on top of its line, like the small intestine. So outer longitudinal is interrupted and forms tenia coli epiploic. So that's why I was telling you that in the intestine, there's a complete inner circular and outer longitudinal. Those are intact. But then in the colon, for instance, the outer longitudinal is interrupted. So that would be the lamina propria. So you can see right from here, you can see that there's a small lumen there. That tells you that this is the epithelial lining, okay, all the way. So it's from the top and then it goes down and then curves and goes up again. You see, because this is so folded upon themselves, you are not seeing much of the pits and its deep parts. Okay, the lumen, you can't see that. And that is how the arrangement goes. In the appendix, we have a transverse or cross section. I told you earlier on that for you to determine that a section is cross or transverse, you have to look at the intact lumen, that the lumen is continuous and intact. You can see the whole entire outline of the lumen, especially if the micrograph has not been truncated, you will find it. Okay. But for longitudinal, you're not going to see anything like that, any circular thing like that. It will be open like this space. So blind sac extending from the cecum You also have crypt only. So usually those kind of epithelia where you have crypt only and no villi or plicae, we describe that as the colonic mucosa. So note the prominent lymphoid nodules. You can see lymphoid nodules there. And this type of lymphoid nodules uh, start from even the mucosa. Okay, so you have lymphoid nodules in the mucosa and you also have them in the submucosa. So lymphoid nodules in lamina propria and submucosa. Lymph nodules disrupt the muscularis mucosa, hence appear as isolated lines of smooth muscle. So because the lymph nodules that are within the lamina propria, and we know the lamina propria is the loose connective tissue of the mucosa, because it's growing outwards towards the submucosa, you end up having it disrupting the muscularis mucosa. Okay, so they will appear as isolated lengths of smooth muscle instead of the continuous lining we find in the small intestine. Muscularis externa has complete inner circular and outer longitudinal layer. But remember the outer longitudinal is interrupted in the colon, but in the appendix, they are complete, both inner circular and outer longitudinal are complete. That is why even in the gross anatomy, you not find epiploic, you know, um, appendices in the appendix. Okay, so you won't find the tinea coli or the epiploic appendix or extensions. So that is how the appendix looks like. So this is also important that muscularis externa of appendix is intact. The inner circular and outer longitudinal is intact. So I put here that compare. If you have this and you need to give them names, how are you going to do that? From the lecture we've had so far, you should be able to tell what this is, what that is, and what that is. Okay, so quickly write them, your answers down before I project my answer, and then you can compare. So I'm just about to show my answers. So that is jejunum. Be sincere to yourself whether you got it right. If you did it right, then kudos. And that is the ileum, and that is colon. Remember the jejunum and its prominent villi and the characteristic nature of it. Okay, they are more like perpendicular villi. Then remember for ileum, you have a lot of lymphoid nodules. Then for colon, they don't have villi, only crypts. But these ones have villi. So you can see that it is going up, and they also have plicae with submucosal core, unlike that one. And also the colon has a lot of goblet cells. I have to add that. Very, very enormous goblet cells. So in summary, we have looked at the duodenum and we said that it has a lot of Brennus gland in the submucosa. We talked about the ileum and we say it has a lot of lymphoid nodules. We also see a lot of lymphoid nodules in the appendix. Okay, so from the distal portion of the intestine, you have most of these containing nodules. So from ileum all the way down. But you don't find those nodules much in jejunum and duodenum. Okay. If you ever find them, they will be isolated. So that is jejunum, the plicae. So basically, that is what we have for the intestines. I want to thank you very much, and I hope this video helps. Thank you.